everyone. Thank you for joining today's session titled The Future of Diagnosis and Treatment in Eye Care. My name is Dr. Yvonne O, and I am Professor of Ophthalmology and Vice Chair of Postgraduate Education in the Department of Ophthalmology at the University of California in San Francisco. I am an Asian woman in my 40s and use she, her pronouns. I will be your moderator for today's discussion. Today, we will explore through a roundtable discussion the connection between eye health and overall well-being, key considerations for genomic research in rare diseases, and innovative device technologies that have the potential to transform the lives of individuals who are blind and visually impaired. I would like to introduce our panelists for this session. We have Dr. Matthew Benson, who is an assistant professor and clinician-scientist in the Faculty of Medicine and Dentistry in the Department of Ophthalmology and Visual Sciences at the University of Alberta in Edmonton, Canada. We have Dr. Yun Liu, who is the Senior Staff Research Scientist at Google Research, and Dr. Philip Choik, who is a professor in the Department of Biomedical Engineering, the Executive Director of the Pritzker Institute of Biomedical Science and Engineering, and Affiliate Professor in the Stewart School of Business at the Illinois Institute of Technology. He is also President and CEO of Cygenics, Inc. So thank you to all of our panelists for joining us today, and we're going to get started now. And to start, we are going to speak with Dr. Yun Liu. He's going to speak about oculomics, the eye as a window to overall health. Dr. Liu, could you introduce yourself and share a little about the work you do at Google Research?
Yeah, of course. Hi, my name is Yun Liu. I'm Asian male. And I go by pronoun he, and I work at Google Research right now. Our team works on using AI to improve healthcare, and we strongly believe that technology such as AI has a lot of potential to help with many things, and one of which is to help improve AI. In terms of the question around oculomics, I think our journey here started maybe a few years ago. So first of all, oculomics is actually a fairly recently defined term. And so probably the best way to think about it is to think about terms like genomics, where it's really about the genes in the body and what you can use it for in terms of detecting disease. So oculomics is really about trying to understand if you can look at the eye and specifically the blood vasculature in the eye. You can use special cameras to look at the vasculature and understand if there are signs of systemic disease. Now, traditionally in the clinic, what doctors may do is that they might take these photos or use a special camera to look at the back of the eye. And what they're looking for are signs of diabetes, which is a disease where the blood sugars in the blood are pretty high. And what they're looking for are signs where these blood vessels get a bit more leaky, and there's sugars and other things that's coming out of the blood that's causing lesions and other abnormalities at the back of the eye that they can then catch. So for people with diabetes, when these abnormalities are seen, they can then be referred to eye doctors for further follow-up and care. Now, what we want to do as part of oculomics is to see if the blood vessels, those same kind of like images that you capture to detect diabetic eye disease, whether you can use that to detect other signs of systemic disease, such as those that's affecting your kidneys, your lungs, or other things. And so our journey here really started also with the diabetic eye disease detection, where we initially built an AI algorithm that can take these images as input and figure out how severe the diabetic eye disease is. And we did quite a bit of work with collaborators in India and in Thailand on studying this AI and to make sure that it's performing really well before we then worked with partners to bring it to real patients in India. So far, it's already helped more than 600,000 patients get screened for diabetic eye disease in India. On the oculomic side is where we were doing sort of like more novel research to understand what the potential is here. Some of our earlier work revolved around using AI to find out if you can see when someone's hemoglobin levels in the blood are low. So one example of that people know about today is if you look at the eye and when someone has very severe anemia where hemoglobin is low, the eyes and other parts of the body may look pretty pale. And so what we showed was that you can actually use an AI to more precisely quantify the amount of hemoglobin in the blood and understand when someone may be at risk of anemia.
I'm curious what advancements in imaging technologies and artificial intelligence have propelled the field of oculomics forward. And the other sort of follow-on question I wanted to hear more about was, are we limited to examining the vasculature, or are there other aspects of the retina that we could learn from?
Yeah, that's a
Also,
great
it's
question.
not going to be
So let me tackle
exhausting.
the second one first because it's slightly more related to our previous discussion. So in terms of
Okay.
what else is in the image actually takes a photo of the retina, of the back of the eye. And alongside the vasculature, you also see the optic disc, which is where the nerves and other parts of anatomy are entering and exiting the eye. And so there's many more things you can see in that image. And in terms of the overall photograph, when you ask AI to perform a certain task, the AI will try and learn what is about these things that it sees in image are actually predictive of the thing that we care about. So this is in contrast to more traditionally when we were working on computer vision approaches, where we would have to hand engineer and really work with experts to say, hey, we want to understand how curvy this part of the blood vessel is. And so you have to figure out ways to describe using numbers and other mathematical functions, like what curvy means for a blood vessel and how do you actually quantify it well. Traditionally, those have been sometimes a little bit brittle, which means that sometimes it doesn't work very well when the image is slightly different, taken from different angle, and so forth. And so with modern AI algorithms, these all become a little bit simpler and definitely looking not just at the blood vessels, but also other parts of the image. The other question was around, what has made all of this possible? And that's a great question. I think there's maybe three primary ingredients. And the first ingredient is data, where we've seen essentially that the amounts of data sets that people have collected over time has increased. And maybe 20 years ago, the same data sets have already started collection, but in more recent years, there's been more patients who have kindly donated their data for retrospective research and so forth. And so these data sets have become larger with time. The second is in terms of compute power and the hardware associated with that. So specifically, computers have gotten faster. Many of you may remember the days where every couple of years you might buy a new computer and it feels very much faster than it was just last year. And so while some of those days are a little behind us in terms of larger scale compute and the ability to train AI algorithms, that progress has actually not slowed. And so we're getting faster and faster and more specialized compute hardware that can be a lot better at computing and training these AI algorithms. The third major ingredient here is really about the essentially the software and the algorithms that we use to train these models. And so some examples of that are one thing I mentioned earlier was how one would describe the curvature of a blood vessel, and that's very hard to do. And so one thing that we have done in sort of like more recent times is to be able to leverage very large what we call artificial neural networks. Now, the way these things work, you can imagine it as essentially kind of inspired by biological neurons in the brain. And essentially what we can do is to have each neuron do a very simple mathematical function. But by assembling many of these neurons, artificial neurons together in many, many layers, we get these very deep kinds of compute structures that can compute very complex functions and do very interesting things. And so it's really by having these more and more sophisticated AI algorithms that we're able to, with time, learn better and better models that can predict things that are interesting and useful for caring for patients.
So it sounds like there's fascinating research going on in terms of developing these AI models to understand and develop this field of oculomics. How do you think oculomics in its current state, or maybe also in the future, can be integrated into routine healthcare to improve patient outcomes and improve health?
Yeah, that's a great question. I think there's a lot of potential here at the high level. And I think one way to think about it is that we want to do more with less, meaning instead of having to do an invasive test, so for example, a biopsy, you would need to essentially make an incision and take out tissue from the body. So that's something that's invasive. There are certain amounts of risks, however minor, associated with the procedure. If you could do more with less by taking a photo of a particular location, especially if you can do it in a non-invasive way, there's a lot of potential in using that photo to derive insights about what else is happening in the body, whether it's about the kidney, the liver, or some other part of the body. And if we know that someone potentially based on that non-invasive image is at risk for certain kinds of conditions, then we can say, well, this person is at higher than average risk. Maybe let's refer them in a very targeted way to get more invasive tests. So in this way, what we can do is really have people who really need the tests, get them in a targeted fashion. You can spare everyone else kind of like the need to do these invasive tests, and that could really helpful from a cost perspective, but overall in terms of the experience of the patient getting screened for these diseases. Now, there's a few ways that one can do this source of disease screening. And one really promising way, I think, is when we move towards opportunistic screening. So one way to think about that is, let's say you are at your eye doctor's for just an annual, exam of the eye, and you're just, checking in. You're not really expecting to find anything. The eye doctor's, doing their part to examine and see if there's any issues. But at the same time that they do it, let's say you're able to get a photograph taken, and you're able to use more advanced technologies to understand if you might be at risk for other conditions. And for those who might be at risk, to then figure out if it's worth, tracking it over time, or going straight to a specialized doctor that can then take better care of you specifically for that condition. So I think, doing these opportunistic screenings in a very convenient way for the patient, at very low cost, and really figuring out who could benefit from more advanced testing could be really a great pathway of the future.
Thanks. How can oculomics be a tool for better collaboration between eye care providers and other healthcare providers? You sort of spoke to that a little bit about potentially linkage of care and doing opportunistic screening, but I'm curious to hear more of your thoughts on that.
Yeah, so I think that this is where, if your care team, which today they already do, but if they can work, very closely as a team to take care of you, where if one thing you notice by, let's say, the eye doctor, then they can refer to another doctor that can then be specialized in that particular condition. So I think, really having your care team, work together as a whole will be really helpful. Maybe adding on a little bit to this is that I think with AI technologies, more advanced technologies, we can maybe start really enabling physicians who are maybe not a specialist in any particular field to be able to help at least do some initial screening for other things, right off the bat. And so instead of relying on the fact that, hey, I need to go see a specialist for every single part of my body, it is possible that in the future you might be able to go to any physician, whether it's for eye or other parts, and that they could, do at least initial sort of like screening to understand if you might benefit from seeing other specialists. So I'm really looking
Yeah.
forward for the future here.
Yeah, I was wondering when you were talking about opportunistic screening, you were talking about an eye care provider picking up other systemic diseases, for example, by looking at the eye. But you could also imagine a scenario where, you visit your primary care physician. And in fact, diabetic screening already happens this way sometimes, right? Do you envision a future where, you go to see your family doctor, your primary care doctor, and then you get some retinal images taken, and then, an AI could help determine if you need to see other subspecialists?
Yeah, for sure. I think that'll be a great outcome where, all of your care sort of like at least the initial phases of that can happen, in one go. And that could really encourage people to seek out care and essentially have a one-stop shop, at least to get started and to figure out if they need to do more.
Great. I'm going to put you a little bit on the spot here, Dr. Liu. I'm going to ask this question on behalf of my daughter, who's a 10-year-old, very interested in climate change and the impact. And I heard this factoid that, every time you ask ChatGPT a question, speaking of AI systems, you are, burning through a bottle of water. And so I'm wondering whether Google
Mm.
research is taking into consideration the impact of these AI models on our environment.
Yeah, that's a great question. So I think we do maybe need to zoom out, I think, in two ways regarding that. I don't actually know specifically for ChatGPT and how much water is being used. I believe the statistics is referring to essentially when these models are being run. These are
Yeah.
fairly large, computation based models, and so they're happening in a data center somewhere, where a data center is
Yes.
essentially a large number of computers. Now, when computers run, you might have felt it with your own personal computer. They generate some amount of heat, and the fans create noise and so forth. So when the heat is created, you can't let the heat continue to climb, or it's not good for the computer, essentially. And so water generally is used to cool the computers now. Now, for cooling purposes, the water is usually recycled for cooling purposes, and so there is some amount that's being kind of like expended and used. But for the most part, the water is sort of like being expended. The other question I think that I get, sort of like very similar to this, is around kind of like overall energy use, and I think this is where we have to, I think, approach the estimates here with a little bit of a grain of salt, where there is energy being used for certain computers to run, But part of the question is, if it was not being run on a computer, like what else would be happening? And so I think there's some very interesting research being done by many talented researchers trying to understand the impact of these, but also recognizing that if you, let's say, were to hire a writer to help you draft out an email, let's pretend we do that for the moment. There's some amount of energy, biological energy perhaps, and also computational energy that's expended by doing that. Versus if you, went to your favorite AI and ask a question and ask it to do something, it expends some energy as well. So what the balance between the two are, that's kind of hard to tell and very difficult of a question to I think answer fully. But definitely this is something that folks care a lot about, and that we are looking closely into. In terms of actual progress, I think what we've seen with time is that when we first started, a lot of these AI models have been even larger than they are today, and with time, these models have become more and more efficient, such that they're able to, I guess, similar to what we discussed earlier, do more with less.
Yeah, that's what made me think of it when you said do more with less. Thank you for entertaining that question.
Yeah.
We're going to move on now to Dr. Matthew Benson, who's going to speak about genomic research in rare diseases. Dr. Benson, could you please introduce yourself and share a little about the work you do around inherited retinal degenerations?
Yes, certainly. And just for those who are visually impaired, I'm Matt Benson. I'm a white male in my mid-30s, and I use a he, him pronouns. And so, yeah, I'm a clinician-scientist and an ophthalmologist from Edmonton, Canada. And so I wear a couple of different hats. I have a clinical and surgical practice. My areas of focus include inherited eye diseases, and a large part of that involves seeing patients with inherited retinal diseases. This could be diseases of the central part of the retina, which is called the macula. Some other disorders affect more of the periphery and cause difficulties with nighttime vision, such as retinitis pigmentosa. So those form probably a large number of the patients I see in the clinic from my genetics practice. And I also manage patients with eye misalignment or strabismus, and that forms my surgical practice as well. And that takes about half my time, and then the rest of my time is spent doing research. And so I have a translational research lab at the University of Alberta, and my group is interested in learning about why specific patients develop problems of the retina, specifically inherited retinal disease problems. And so we have sort of a neat approach where we take samples from patients. Usually patients are interested in research. We consent them to participate in our studies, and we collect skin samples, and we grow these cells in the lab and then reprogram these cells to stem cells. And then we take the stem cells to retinal sulfate. So essentially, we can grow the patient's cells in culture and model their retinal condition without having to take a biopsy, like Dr. Liu said, would be quite invasive. So we get around this problem this way, and then we can study the patient's specific disease in culture. And I also have a kind of a smaller aspect of my research is clinical research, and we're involved in a few clinical trials as well. And these are related to patients that have inherited retinal diseases. So yeah, all of that keeps me fairly busy.
Yes, definitely. So, as a clinician, putting your clinician hat on first, what are the primary obstacles in diagnosing those rare diseases, and how has genomic research addressed these challenges?
Yeah, no, thanks for the question. I think probably the biggest barrier that I've come across is just education and awareness on the side of the providers and patients, that we now have access to tools that can allow us to provide a very specific diagnosis for patients with rare diseases. Most of the time, if a patient's seen by an eye care provider or an ophthalmologist, you generally can get a sense that there's a rare disease or something that's not commonly seen, in patients that come to your practice. And so I think that that first step in suspecting that there's an inherited retinal disease or an inherited eye process or some sort of rare eye disease is usually the provider's usually aware of that. But then often, and for a number of years, we've been in this space where we haven't had good treatment options for these patients. So potentially they were told, nothing can be done, and then patients may not necessarily follow up regularly for their eye health checks, or they just have been labeled as having a condition such as retinitis pigmentosa, which I refer to as this inherited retinal disease that causes difficulties with peripheral vision and nighttime vision. And so they may have been given that clinical diagnosis, and that's that's kind of where things had been left at. But nowadays, because of genomic research efforts, we're now able to provide very specific diagnoses for patients. And we now know that retinitis pigmentosa actually includes about 80 different genetic subtypes, and they're all a little bit different. They can be inherited differently and have different implications for patients and their families. And so just with increased tools in our toolbox from genomic research efforts, we're now able to provide very accurate diagnoses for patients. And it's maybe been over the past 10 or 15 years, this has become more and more standard of care. And I would say essentially now if patients do have access to a center that can offer genetic testing, which is becoming more widespread, I would say it would be the standard of care. And so now potentially the barrier is shifting to, finding specialists that see patients with these rare conditions and reaching out to the providers that they're currently seeing to advocate that there's more that can be done. And even though a lot of these rare diseases are still, unfortunately, in a place where there's not good treatment options, there's lots of research ongoing. And by obtaining a molecular diagnosis, by knowing exactly what the change is in a gene that's causing the specific problem, puts patients in line for potential clinical trials and very precision medicine approaches to try to treat these conditions. And so I'm very optimistic about the future, and genomic research has really been driving this and providing the tools for us to advance the field. And we do have a gene therapy that's approved for one very particular type of inherited retinal condition. And essentially, in this particular condition, patients are missing a particular gene. And there's a surgical approach where you deliver that normal copy of the gene back into the retina to boost vision and prevent it from getting worse over time. So that was something that potentially would have been thought of as science fiction, 20 plus years ago. But these therapies are becoming a reality, and it's all been driven through research efforts. And just taking along off of what Dr. Liu was mentioning about AI, I came across a recent software. It's more in research development right now, but it's called Eye to Gene. And the idea is that you can take a fundus image or a picture of the back of somebody's eye and even get a sense of what gene may be implicated for their condition if it's an inherited eye disorder. And the system will, I think, give you kind of a rank list of the likelihood of it being, say, this gene causing retinitis pigmentosa versus another. And so, I might be out of a job fairly soon too from AI, because the system's getting very good, and there are lots of tools that I think are going to be very exciting and coming to the clinic very soon.
That is really exciting. My specialty is glaucoma, which is considered also having elements of a genetic disease, although in a different way than, say, inherited retinal degenerations like retinitis pigmentosa. And the field has wanted to move, I think, more towards gene therapies that are gene agnostic, meaning they're not necessarily targeting a specific gene mutation, but rather an approach in which no matter what mutation you might have, the strategy might be helpful. And so I'm wondering, are inherited retinal degenerations amenable to that strategy? Are there groups trying to go after a more broad-based approach?
Yeah, it's a very good point. And maybe for the last 5 to 10 years, there's been more of a drive on developing very specific therapies for patients that have very specific changes in genes. But what some of the difficulties that the field is encountering is that there's such a small group of patients that may be eligible for something like this. And it's difficult to find patients that have these very rare conditions to even enroll them in clinical studies to see if the therapies will be successful. And so there are certainly big efforts being made at the moment to try to develop what we call gene agnostic therapies, where it's still a precision medicine in that you're still treating the underlying pathway that's wrong. You're just maybe not treating that specific change in the patient's gene. And so there are a number of clinical trials ongoing looking at this specifically for inherited retinal diseases. There's a few different strategies. It's all very much the same concept where you're delivering a gene, except the gene is not necessarily the same gene that the patient is missing, but a gene that may be important in modifying that pathway and potentially rescuing some vision. And so I came across at a meeting recently this term. We've heard about precision medicine, which is kind of developing very precise therapies for patients, but it's almost imprecise medicine. It's not really that, but kind of taking a step back and seeing how can we broaden these therapies to more patients so that it doesn't become cost prohibitive to develop all of these treatments for everybody.
Yes. With regards to data sharing, I have a question about how researchers can balance the need for data sharing in genomic studies with the imperative to protect patient privacy and prevent possible discrimination or misuse, especially in small patient populations that you alluded to.
Yes, this is a common concern that patients have, and certainly is understandable. I can kind of answer this in two different ways, more clinically and then also on the research side. I work very closely with a genetic counselor who helps address these concerns with patients that we're doing clinical genetic testing for. It's always it's something important for patients to consider is who would have access to their genetic testing results. If it's done on a clinical basis, of course, the healthcare providers would have access to it, but it really depends on the state and even country in terms of which third parties might be allowed to ask about the genetic testing results or potentially have access to them. And so I would encourage anyone considering getting genetic testing for their condition to have these conversations with their genetic counselors, because they would give them more specific information about what may be which elements of their genetic testing information may be reachable by third parties, because it depends on the jurisdiction, but it is a very important consideration. From the research side of things, I would say that one of the safeguards is the research ethic boards that are associated with, you need for any human studies, you need to achieve, obtain an ethics approval in order to proceed. And they're very strict about only including the minimum amount of patient related information that your study would require to address your outcome. And so this also applies to genetic information and avoiding the use of multiple labels like patient name, date of birth, and age, and these sorts of things. And so instead, you'll apply a patient identification number that anonymizes the data. But the genetic information would still be there. And then the question becomes, well, who would have access to this as part of a research study? And this all has to be very carefully outlined in research protocols where this information has to be stored in a very particular place that's password and password protected. It's encrypted. It's only accessed by very study folk that are directly part of that research project. And if there is any data sharing with other universities, that all has to be well documented as well. So there are many, many safeguards as part of the research on the research side of things. But I would still say that, things can slip through the cracks, unfortunately, despite our best intents. And so it is still an important consideration that whenever you're participating in research, there's always a possibility that that some of these results might might not stay within the research team, although it's highly unlikely. It's still something that if patients are considering participating in research, they should ask the study coordinators and the research teams about how their results will stay within that study, and who would potentially have access to their results. So I think it's a very good question to ask.
Yeah, always important to ask questions. What strategies can be employed to involve patients and advocacy groups in the design and implementation of genomic research studies for rare diseases?
Yeah, that's a great question, and I'm kind of part of clinical research, also basic science research, and I'd say in both fronts, we try to get patients and patient advocates and policy makers involved in the research as much as possible. This even starts with when we write grants, which are essentially a proposal where we have a budget and we're requesting funders for money to support our research and the particular project. A lot of these granting agencies are requiring that we have a knowledge user, is what they call it. It's essentially a patient or a policymaker or somebody who would be able to act on the research findings as a member of the study team. And so on our federal funding applications, the knowledge user is someone that's encouraged to be included in our research projects. And I think that provides a nice way that patients can be directly involved, or policymakers, in the research from the very start, and not just on the implementation side of things. And then in terms of how patients might and policymakers might become more aware of what's going on in research in general, I think becoming involved in organizations like the Foundation Fighting Blindness, which is the US equivalent, or the Canadian equivalent, Fighting Blindness Canada. These organizations can allow patients to stay up to date on what is out there in terms of research, and potentially even link patients to ongoing research projects, whether that's a clinical trial or a natural history study or something like that. And many of these organizations also have what's called a patient registry. And so they include different degrees of information. From the Fighting Blindness Canada standpoint, it's predominantly just the genetic information, but the patients would essentially enroll in a large database. Again, you want to ask questions about, who has access to this information, but it's also vetted by a research ethics board. And the idea is that then you've got your information down so that if there was any studies that came out that might be relevant for you, then there's already a path forward that you'd be made aware of trials that you may be interested in. And so that's one of the difficulties, is that these are rare conditions, and the patients are not always seen, patients come to me from kind of all parts of the country, and I don't always get a chance to see them all the time and keep them updated on the research. And I may have seen a patient, say, 5 years ago, and then it's always one of these things where I'm not sure if they would want me to just call them out of the blue and tell them about a study that they might or might not be interested in. But this is one way that they can kind of raise their hand and say they're interested in staying informed. So I'd say the patient registries are a great way for patients to get more involved in the research world.
That's really helpful. I will also, maybe we could take just a minute or two also just to discuss how to evaluate a clinical research study for scientific validity, or ways in which to avoid being in a situation that may not be as positive as one would hope. I guess what I'm referring to really is, clinicaltrials.gov, which is United States-based site. Those, I oftentimes, patients will talk to me about studies that they've identified on that website. And what I will tell them is that being on that website does not mean that the government or any kind of agency has necessarily approved that study. Presumably, it's self-reported whether an ethics board, as you were kind of referring to, or a regulatory board has evaluated that study. But I always tell patients a couple of things. One is to ask a trusted physician or clinician about whether or not the scientific basis of that study makes sense. You certainly should not be paying money to participate in a clinical research study. In fact, if anything, you may be paid, in terms of, providing parking expenses or other types of financial incentives sometimes are included in clinical research studies, just to ensure that the researchers can kind of help you. You're contributing your time, your efforts to the research project. And do you have any other sort of important advice that you give to patients when they're evaluating studies?
Oh, I think those are all very important points and all excellent. And it's what certainly I would recommend. And it starts with the discussions with the patients and the providers. You don't want to get yourself as a patient into a position where, there's a study that's trying to recruit you and you don't have an opportunity to maybe ask somebody who is more removed from that relationship. I guess just on top of that, of what you mentioned, and maybe the co-panelists can add their thoughts as well. But I've come across it's a difficulty I face myself too. Is once you've kind of described the study to patients and you let them know about the risks and benefits, it's still very difficult to be entirely neutral about a study. And this is, say, for example, one that I'm not involved in, regardless, I suppose, but especially I'm just thinking about one that I'm not involved in, because you kind of, in your mind, you have a sense of which patient might be the ideal patient for that study, who, the study's ideally designed to see if a therapy is going to be effective, but the patient shouldn't necessarily go into the study thinking it's going to be a cure, because a lot of the studies, unfortunately, are not that way, and we're just learning more about how we can develop treatments. And so you kind of get a sense of which patients might be better suited for a study, even though multiple patients may meet the inclusion criteria. And so you kind of have to keep in mind your own biases about whether someone is keen or not to participate, because what might seem low risk to me would be seem high risk to somebody else. And a lot of the decision to participate in trials is kind of value based, and there are other personal factors that go into it too, and that is sometimes difficult to navigate as a healthcare provider. And often once you've provided sort of the scientific basis for the study to the patient, it's helpful for them to touch base with, maybe family members and friends, and get their thoughts on whether it makes sense to participate in a study too.
Thank you. Okay, I'd like to turn it over to Dr. Philip Troik, who is going to speak about innovative device technologies transforming the lives of individuals who are blind and visually impaired. Dr. Troik, could you introduce yourself and share a little bit about your work in this area?
Sure. Thank you. I'm Philip Troik. I am a professor of biomedical engineering at the Illinois Institute of Technology. I'm white male with dark hair, glasses, and although anybody seeing this sees a background that says prevent blindness, I'm really sitting in the basement of a hotel because I'm at what's called Rehab Week in Chicago, which is a collection of 5 different societies that is involved in different rehabilitation technologies, particularly for things called functional electrical stimulation, which is related to the topic we'll have today. My work is in what is called neural prostheses or neural interfaces, and that's just a complicated name for electronic gadgets that interface with the biological nervous system in the brain, in the periphery, or even for organs. And the electronic devices compensate or mimic natural biological function. And in the case of vision, these are called visual prostheses. And they are basically a technological way to take artificial vision information and introduce it into the biological vision systems. So these can operate at the retina level, where with electrical stimulation, with a group of very tiny electrodes, tiny electric currents, will stimulate the residual neural function in the retina, or in the optic nerve, or at the brain itself. So if you think of it that, we're trying to find the point at which the vision system has a deficiency, and then we're going upstream of that toward the brain, and we're trying to take information that would be captured by a camera and then artificially communicate it to the brain in the hope that the brain can make some sense out of it, and thereby producing what we call artificial vision. So this field has a long, rich history, and it dates back really to, from a prosthesis standpoint, to the late 1960s, in which in the United Kingdom, there was the implantation, rather remarkable, of electronic packages and electrodes for a brain-based vision prosthesis. And despite some technological limitations, it was really a groundbreaking study. And in the US in the 1960s, well, US thought if somebody else did it, we can do it a lot better. So, the Russians went to the moon, we can go to the moon. We're trying to go up into space, we can go to the space. So the National Institutes of Health started a project to do a microstimulation of the brain with tiny penetrating electrodes that would actually penetrate the brain surface. These electrodes are so tiny that on the tip of a hair, you could put 10 or 20 of them. So they're very, very small. And the idea is that you would stimulate the neurons in the brain that are involved with vision, thereby communicating information, vision information from a camera directly to the brain. So this all sounds very futuristic, and there was a show in the 1960s, 70s called The $6 Million Man, in which the word bionic sort of came into the public use. But it's pretty difficult to do because we don't really understand the language of the brain. And I often say, imagine if you just opened up a computer and threw a bunch of wires inside and tried to get your Windows operating system to come up. That's sort of what we're trying to do, except we're relying upon the immense sophistication of the brain to make sense out of very limited information that we would put in.
So thank you for explaining to us some of the history of this field. And I am curious to hear more about your visual prosthesis clinical trial as a novel assistive technology and where you see these assistive technologies and the field of visual prosthesis going in the future. Where are we now and where are we going?
Yeah, so we've been working in a team-based project, of which it's really been a privilege to lead this team for about the past 25 years. And the goal of it was really to be able to create an implantable system in which these micro-sized electrodes could chronically be placed and implanted, and we could test the interface to try to figure out how we could use it for artificial vision. So we developed this technology funded by a variety of sources, National Institutes of Health, the Army, and now under the Brain Initiative, in which we have very small implantable stimulation devices. And these are about the size of a button that's on the top of an AA battery. So they're very small. They're about 5 millimeters in diameter, if that means something to people who are metric. But it's about the size of the little button that's on an AA battery. And coming out from that little disc, there are these 16 of these extremely tiny hair-like electrodes. And this little device, which is called a wireless floating microelectrode array, gets inserted into the brain. It looks like a little miniature hairbrush. And these little electrodes get stuck in, and each one is completely wireless. And so it's powered wirelessly from outside the body. And you can communicate with it. And with a collection of these in your head, it's like a little cell phone network in your head. And each electrode has its own phone number, and each module has its own area code. And so we can call up any electrode wirelessly and tell it what to do. And so then the question is, what do we tell it to do? So we are funded by the Brain Initiative, thank you very much, NIH, and we have now implanted 2 volunteers with these modules. The first one has 25 of the modules, totaling 400 electrodes, and the second person has 32 modules, totaling 512 electrodes. So to people in my field, that sounds impressive, but bear in mind that in the vision, the occipital lobe on each side of our head, we got about 350 million neurons. So we're far below what biological vision might be. But when you stimulate through these, the person perceives what they think is a spot of light. Now, it's not a spot of light because light hits the eye and gets converted by the retina, but it's the perception of a spot of light because we're stimulating the brain and we're really manipulating the vision perception machinery that's there. And so the idea is that with a collection of these spots, you could then have a crude form of artificial vision. So it's important to realize that where is the field right now for this? And there are systems like this at the retina. There have been some at the optic nerve. Ours is at the cortex. Ours is unique because it is completely wireless. There are no connectors through the skin, no wires leading to the brain. So it's revolutionary in that sense. And it's made to be chronic, which is a big advantage because then you have time to work with the person to see what could they achieve. So our person now is our longest one that's been in for 40 months. And so, and the technology, I'm gratified to say, is working extremely well. But what might you be able to do with this? That's unclear, and that's what we're trying to find out.
Well, listening to you explain your technology makes me realize that one of the projects in my lab will be completely made obsolete, Dr. Troik, by your visual prosthetic because, we're trying to treat glaucoma by transplanting stem cell-derived retinal ganglion cells. But of course, we have to figure out how to get those ganglion cells to rewire to appropriate synaptic partners in the brain. But your system will bypass that entirely and is very promising in that way in terms of, for the transplantation field. If we can bypass it completely, maybe that is the better strategy to go.
Well, I hesitate using the word better for any things. It's different.
Yeah.
And so in uh, for the technology for a cortical vision prosthesis, of course, the person doesn't need physical eyes. And in fact, one of our participants has some enucleation of eyes. And so naturally, you're bypassing the eye and the optic nerve. You're going directly to the brain. So yes, in concept, that's true. The difficulty is knowing the language to talk to the brain with, especially with the crude interface we have. So imagine if you took a kitchen colander where you strain noodles and you held it up in front of you with these little dots and you blacked out some of them and you kind of moved it around and you tried to see what was in front of you. That's a little bit what it's like. But
Yeah.
certainly the participants that we have can do some surprising things. And our participants right now need to have no light perception or bare light perception. So, what does that mean? Well, if you can see a hand waving in front of your face, it's too much vision. And that makes sense in any kind of these trials because you don't want to risk compromising whatever residual vision might be there. But at the same time, it's giving us the opportunity to learn about how we can do this interface. And we are looking for additional participants for the trial, and we do have a process. I think there is going to be an email address put up here about where people can contact us. It's very easy because it's our trial is the intracortical vision prosthesis, so it's icvp@iit.edu. So it's very simple email address. So we do respond, but the idea of competition, I often get asked that, well, why did you choose the brain instead of the retina? Why not the optic nerve? Why not the thalamus? Why not? Well, people choose to work on things because you find a kind of get in the groove and you're starting to do that. So I don't see things as that competitive. I see them as different. And I see that we need to explore all modalities of technology to try to reach what we all imagine through science fiction, which is one day it'll be standard of care to be able to have an artificial vision system, whether you're a Star Trek fan or whatever, that can replace the biological function. And, this is like the cochlear prostheses. For cochlear implants for hearing, those are extremely successful. The interesting thing about that, though, they were successful when they only had 2 electrodes. And that just tells you that it's a lot easier to interface to the auditory system than it is to the vision system. As I'm sure you know, almost half the brain uses vision in some form or another, whether it's by mapping memory, whether it's by... So there's a lot of neural machinery there that's involved in vision processing. And we're really only at the baby stages of being able to do that. But it is still very gratifying that this work one day will translate into that. And these types of devices are really the first positive step to being able to have this chronic interface.
No, I absolutely agree what you said about competition. All the different groups of scientists, all the teams that are studying different aspects all along the visual pathway, I think are critical to us all being able to develop technologies that help patients. And, it just, like you said, it speaks also to the complexity of the visual system. When you think about also just all the different visual processing that is happening in the retina and then also in the brain. I know you've already sort of alluded to this, but maybe you could speak a little bit more about the impact of patient and caregiver involvement in the development of assistive device technologies and why it's critically important.
Yeah, so anybody who works in medical device design knows that it's not optional. It's a requirement that the design process be able to demonstrate that it had input from what we call user needs. And so, the phrase, nothing about us without us, is really key to the development of any technology that's used in a medical device, in which the potential users, whether they be clinicians or individuals with blindness, that they have input into the process. And in our case, our participants are actually members of our research team. That is how they are regarded. They make suggestions about doing tests. Without them, we can know nothing, because they're the ones that are helping us go to a place that we haven't been before, because they can tell us what they perceive when our technology stimulates their brain. And so it's absolutely essential that that the users be considered as really part of the research team. We hate the words human subjects. We hate that word because it degrades and really denigrates the volunteerism and the sacrifices that people are making. They're putting themselves at risk in order to help us advance knowledge to benefit others. And so but I'm often, I have to say, disturbed by hearing many of my colleagues not consider the brave people who are volunteering for studies to really be those who are enabling our work rather than just making it convenient. And so, often in our field, it was said, well, we have to have human studies because animals can't tell us what they see. And so my response was, well, so you want to use people because they're smarter primates. That's the reason, right? It's not that there's any real sense that they are actually participating in our work.
Thank you. We're going to move into a little bit of Q&A from the Q&A function in the webinar. So the first question is for Dr. Troik. Is the trial open for children or adults only, or both?
No, our trial is for people between the ages of 18 and 71. And there are special issues for consent for children in studies. And above the age of 70, there is a higher risk because of the cranial surgery that has to take place.
Mm-hmm. I also wanted to ask a question that I know Dr. Liu answered in the webinar session chat part, but just in case people missed that, we had a question about, is there any group that is developing an AI model to diagnose retinopathy of prematurity?
Yes, for sure. There's a number of researchers in that general area. One example is the folks in working with Michael Chiang. He's now running the National Eye Institute, so maybe slightly less research activities these days. But he has a sort of like a fairly extensive publication portfolio with research work that he and others have done in that space. Here's one example of several researchers in this space. I've dropped a keyword if you'd like to learn more. At some of the time that this work was done, the kind of AI that was used was something called deep learning. So if you look for ROP with the deep learning kind of like search term, that should bring up a number of relevant studies.
Thank you. Dr. Troik, there's a question about whether or not, about asking about the work of Dr. Sheila Nirenberg, who has worked out algorithms that mimic the brain's interpolation of visual stimuli.
Well, I'm not directly familiar with that work. One does have to be careful about the idea that if we artificially stimulate the brain, that we are mimicking natural biological processes. In fact, the artificial stimulation of the brain is done in a completely non-biological way. It's really a different language that we're trying to teach the brain to interpret from a visual standpoint. So while knowledge about how the biological vision system functions is helpful, it's often not enabling to understanding what we do artificially when we try to activate those same neurons.
Thank you. I have another question for Dr. Liu. Is there an AI model to evaluate strabismus and ocular motility?
There probably is. It's not an area I've looked into myself or as much. I think for some of these, they perhaps do more of the what we think of as the front of the eye as opposed to back of the eye, and so it's like a slightly kind of different approach might help. On our end, some of the research we've done, we call it the external eye just because it's the outside of the eye, and so we have done some work with external eye images to look at both systemic as well as diseases of the retina or the back of the eye. not these two specifically. I am mostly because I think in terms of feasibility, the data that we're working with were retrospective and already collected. Those data were still images. So I think for some of the ones that you mentioned, these might be more feasible if you had videos of the eye. And so far we haven't, had found, I guess, good data sets that contain those. That's a great question, though.
Yeah, I think it's an area, I'm sure there probably is some research teams working on this, because it is an area that you could imagine would be very ripe for developing those models, because the photographs are fairly also, and videos would be, fairly straightforward to obtain. Okay, I am going to now move towards, I want to just first thank all of our panelists and all of the audience for joining us today. So now I'm going to hand it over to Jeff Todd, President and CEO of Prevent Blindness, to provide today's closing remarks.
That was just a terrific discussion, and I just thought that the entire day, I was just wowed by the breadth of the conversations that we had throughout the day. And I'd like to thank our presenters, our moderators, our summit sponsors for their support, and you for joining us in celebrating 14 years of critical discussions on vision and eye health. And finally, and maybe most importantly, please sign up for our advocacy alerts and support the EDV Act. Together, all of us can make sure that children's eye health becomes a legislated national priority. We look forward to seeing you next year. Have a wonderful day.